Good day. I'm Brian Hoffland, President of Research to Prevent Blindness. Welcome to RPB's Lunch and Learn on Ocular Genetics. Research to Prevent Blindness is a leading nonprofit organization supporting eye research directed at the prevention, treatment, or eradication of all diseases that destroy sight. RPB was founded in 1960, and over our 65-year history, we have awarded more than $425 million in research grants to the most talented vision scientists at the nation's leading medical schools. As a result, RPB has been associated with nearly every major breakthrough in the understanding and treatment of vision loss. We are incredibly proud of the breakthroughs made by RPB-supported researchers, but there's still so many treatments that need to be developed, so our work continues to fund the breakthroughs of tomorrow. In today's educational session, you will hear from two RPB-supported talented scientists about ocular genetics and its potential for creating treatments and about cutting-edge research. Both of our speakers, Dr. Jeremy Nathans and Dr. Jean Bennett, have received past RPB awards. We thank all of you for the generous supporters of RPB who make our work possible. I'd especially like to thank Johnson & Johnson for their support of today's educational event. The importance of supporting medical research has never been so apparent. We thank you for your partnership in supporting innovative vision research to ensure that new breakthroughs are possible. If you have not yet supported RPB, please consider doing so to help make sure that we're able to continue with these breakthroughs. But getting back to today's session, let me introduce our moderator. Robert B. Hufnagel, MD, PhD, is a physician scientist who joined the Kaiser Permanente Center for Integrated Health Research in Hawaii in 2023. He received his MD and PhD from the University of Cincinnati. He completed his pediatrics and clinical genetics residency at Cincinnati Children's Hospital, and he's also completed fellowships at the National Eye Institute and the National Human Genome Research Institute. His research is dedicated to improving utilization of genomic data in medicine at point of care. His projects combine clinical data, biostatistics, and high-throughput sequencing analysis, all to establish patient-centered disease models for translational and preclinical studies. Dr. Hufnagel serves in leadership roles in several clinical and research organizations. As moderator, he will introduce our two speakers for today and moderate the Q&A session at the end. Each speaker will speak for 20 minutes, and I will serve as timekeeper. I will give each speaker a warning at the 18-minute mark. We just want to make sure that we have enough time at the end for a robust Q&A. And with that, Dr. Hufnagel, please take it away.
Hi, thank you so much, Brian, and thank you for the opportunity to moderate this excellent session. And, our event today is about ocular genetics, and our two speakers are physician scientists who inspired me to become a physician scientist when I was in my training. And over the past 40 years, the field of ocular genetics and genetics in general has seen a very robust acceleration in being able to provide molecular diagnosis for patients who have blinding diseases, as well as enabling then treatments for those diseases as we understand the mechanisms and understand how to treat those mechanisms, in large part through gene therapy. Both of these big advances have really allowed people to progress through their diagnostic odyssey, as many people wait a long time to get the answer for why they have progressive blindness. As well as a treatment and therapeutic odyssey, as we have now FDA-approved gene therapy and more, hopefully, on the horizon. Now, I'm pleased to introduce our first speaker, Dr. Jeremy Nathans. And as a personal anecdote, he wrote two of my favorite back-to-back science papers uncovering the genetics of why we have color vision and why color vision is important in normal sight. He's a professor of molecular biology and genetics, neuroscience, and ophthalmology at the Johns Hopkins University School of Medicine, and an investigator in the Howard Hughes Medical Institute. He received undergrad education at MIT, MD and PhD degrees from Stanford University School of Medicine, and postdoctoral research at Genentech. He's published many, many papers, over 200, including research papers, book chapters, and review articles, and received numerous awards for his research. He's also an elected member of the National Academy of Science and the National Academy of Medicine, and serves on multiple advisory and grant review boards. And for the past 25 years, he's been a member of the editorial board of proceedings of the National Academy of Sciences. He's currently the chairman and scientific advisory board of the Klingenstein Simons Fellowship Award in Neuroscience. And over the past 35 years, his laboratory is focused on the intersection of human genetics, ophthalmology, retinal cell biology, developmental neuroscience, and evolution. And the lab's current focus and topic of the talk is on vascular biology and disease, with a special emphasis on the blood-retina barrier and the blood-brain barrier. So I'd like to introduce Dr. Jeremy Nathans, MD, PhD. Looking forward to your talk.
Rob, thank you very much for that generous introduction, and my thanks to the entire RPB team for hosting this event and for allowing me to participate, inviting me to participate. I want to tell you about work we've been doing recently and also the larger picture regarding vascular disease in the retina. And let's just start in the next slide. Val's going to advance the slides for me with a picture of the eye and the position of the retina at the back of the eye. I think you can see in this cross-sectional view how light enters on the left side. It's refracted at the cornea and then by the lens, and then the image is formed on the back wall of the eye, which is lined by a thin layer of neural tissue called the retina. It's shown in yellow here. And on the next slide, we see a microscopic image of a primate retina. That this is very much like our own retina. The light in the way this image is set up on the slide would enter from the bottom and proceed through the thickness of the retina. It's about a 20th of an inch in thickness. And then the light is absorbed by these little green cone-shaped objects at the very top, which are the regions of the photoreceptor cells with the light sensing receptors. Now, not shown here, but important for this talk, is that there are blood vessels that feed the retina. I think there might be a few in this image, but they're not stained, so they don't stand out much, but we'll see them in the future. And they, although not numerically a large part of the retina, are critical for its function because, like all tissues in the body, the retina needs a blood supply. It needs oxygen and nutrients. And the beginning of the story, I would say, really goes back to the middle of the 1800s, the 19th century. And on the next slide is one of the heroes of this story. This is Hermann von Helmholtz at a time in his late 20s when he was just beginning his medical practice. He has an MD at this point, and he was fascinated by optics and by the eye. And he was aware that the interior of the eye was at that time completely terra incognita to the medical community. And we all know if you look at someone's pupils, that is the entry point, the openings to the eye, the pupils look black. The light goes in, but almost no light comes out. So you can't see what's going on inside. And Helmholtz set about devising a device that would allow an ophthalmologist or a physician at the time, I think it's fair to say it was any physician, to look inside the eye. And the next slide shows in schematic form that device. It's now called the ophthalmoscope. And on the right, we see the doctor's eye, and on the left, the patient's eye. And the trick, the Helmholtz trick, was to use what's called a half-silvered mirror, a mirror that allows about half the light to pass through it and half to bounce off. And it's placed at a 45-degree angle between the two eyes. This is inside of an ophthalmoscope when a ophthalmologist looks into your eyes holding the handheld ophthalmoscope. And a light source shown coming from the bottom bounces off the mirror. It then, at least about half of the light, goes to the left into the patient's eye. So this is a critical part, a very strong light source. So there's plenty of light going into the eye, and a tiny fraction of it bounces back out again. Tiny because almost all the light is absorbed, but enough is bounced out of the eye and heads back through the mirror, and some of it goes through that mirror because it's only half silvered, semi-reflecting, in other words, that some of that light ends up in the eye of the physician, and that's the eye on the right, and that person can see inside the patient's eye. Now, on the next slide is a photograph of what the ophthalmologist sees. And this is the retina, the surface of the retina of a human. The blood vessels are obvious here. They emerge from the right side, what's called the optic disc. They circle above and below the center of the retina. And in the very center where fewer vessels, or at least fewer large vessels, are seen, that's the region of highest acuity vision. That's the most valuable part of our retina. So this is basically where things stood as of the 1960s or 70s or 80s. But in the 1990s, a very important invention, the next big step after Helmholtz's invention of the ophthalmoscope, was made. And on the next slide is a picture of that device. It's a method of visualizing the retina called optical coherence tomography, OCT. And here's a patient on the left getting this scan. It's very rapid, takes less than a minute, completely safe and non-invasive, and the images are shown on the computer screen of the device. And the critical advance here, and that's shown on the next slide, is that the image that the ophthalmologist gets from OCT is a cross-section of the retina. They're not getting an image only looking down on the face of the retina, the classical ophthalmoscopic image. The OCT is a little bit like radar or like ultrasound. It has a penetration of tissue. It's using infrared light. The light goes into the retina and it bounces off the different retinal layers. And like radar, the light that has bounced off can be picked up from a detector. And here what we're seeing on the top half of this image is a histologic section, a section through an eye obtained after the death of the individual. And at the bottom is the optical coherence tomography image of a typical human retina. And the central region, which is depressed, is that region in the center of the retina, the so-called fovea. So this turns out to be enormously useful to ophthalmologists. And it has revolutionized the diagnostics of retinal disease. Now, I want to just introduce one more technique. It's on the next slide. And that is a method in which one looks through an ophthalmoscope, again, like the kind that Helmholtz invented. But one doesn't just look at the appearance of the retina from the illumination of white light. One looks at the fluorescence of a dye that has been introduced into the vein of the patient. So a sample is injected into a vein in the arm. The dye goes everywhere in the body through the circulation. And shown, let's just focus on the left three panels, A, B, and C. If we look at different times, we see that initially, that's the top left panel, the major blood vessels light up. These are the arteries lighting up first. And then in B, we see additional vessels. These are now veins lighting up. And in C, yet additional ones. And in addition, there's a little arrow pointing to a region where there's been some leakage of the dye out of the vessels. And this is an abnormality and should not be present. So this indicates a vascular defect in this individual. So this method called fluorescein angiography or indocyanin green angiography, depending which dye is used, turns out to be incredibly powerful for determining the structure and the integrity of blood vessels in the retina. Okay, now let's move to the next slide and just step back a second and talk about vasculature, because that's really what we'll talk about for the rest of this little talk. This is a hand drawing from the 1500s from Vesalius of the human vascular system. Of course, we learned about this in high school. The vasculature is basically the plumbing of the body. It goes everywhere. And if on the next slide we ask what controls the properties of the vasculature, it turns out there's a master regulator. It's called VEGF, vascular endothelial growth factor. It's shown in that little cartoon as two little balls connected together because it's a dimer. It's a pair of molecules joined together. And these are proteins, and they're made by tissues, and in particular by tissues that aren't getting enough oxygen and nutrients. They produce VEGF which binds to a set of receptors on vascular cells, and they cause the vasculature to proliferate. EC in this little cartoon stands for endothelial cell, the cells that make up the blood vessels. It causes those cells to proliferate. It causes them to migrate. And strangely, we don't know quite why this is so, it causes them to increase their permeability, that is, to leak liquid content from the blood space into the tissue. That last one turns out to be a very big deal in ophthalmology. And on the next slide, I want to just show you with the optical coherence tomography method, what it looks like to have a retinal vascular disease of the sort that many patients with diabetes have. It's called diabetic macular edema. And in the upper panel, so there are a series of 3 panels, they're from the same patient, taken at yearly intervals. The upper panel is this diabetic patient before treatment began, and the retina shown in cross-section to the right shows a series of black spots. These are actually cysts filled with fluid, which are distorting the retina, enormously distorting it. The retina is a bit like a sponge, and it can soak up a lot of liquid, but that is not good for the retina. It's damaging to the retina, and it degrades vision. And this person was given as an intraocular injection, that is an injection into the eye, a medication called an anti-VEGF monoclonal antibody. It's given typically once a month or once every 2 months. And 1 year later, in the image in the center, I think you can appreciate that the cysts have shrunk, the retina's decreased in size and thickness. And 2 years later, at the very bottom, the cysts are essentially gone and the retina is a normal thickness, and vision has been restored. So this is a remarkable treatment. It came online about 20 years ago. Two American companies, Genentech and Regeneron, are the major players in this area. And this has restored vision to many hundreds of thousands of patients. But I'll just say, despite its many successes, there are many patients who do not have this sort of a very positive response. About 50% of patients have less than optimal responses, and many of them really have very minimal responses, if any at all. So there's room for improvement for many, many patients. So now if we go to the next slide, this takes us to genetics. Mette Warburg, an ophthalmologist in Denmark who died about 20 some years ago, was really the pioneer, and she described a set of families, one of which is shown on the next slide, or I should say their family tree is shown on the next slide, in which there is an inherited defect in the development of the blood vessels in the eye. She named it Norrie disease after her mentor, Gordon Norrie. And on the next slide, I want to just show you what a retina with vascular disease, one of these inherited vascular diseases, looks like. This is actually a milder form than Norrie disease. Tragically, the children with Norrie disease, or individuals of any age with Norrie disease, are completely blind. The vascular deficiency is so severe. But there are others, and an example is shown here of someone with a milder vascular deficiency, and that's the retina we see on the right. The left retina is a normal retina. The one on the right is from a person with a disease with a very long name, familial exudative vitreoretinopathy. It's an inherited disease, runs in families, as the first word suggests. We call it FEVR for short, by its initials. And on the right side of this image, I think you can see the retinal fold. The retina has been detached in that region from the back of the eye. It's severely damaged, and that's secondary to its lack of a blood supply in that region. So this is a defect where part of the retina has a normal blood supply and part is deficient in its blood supply. Now, I think we can go to the next slide. Probably we'll skip this one. Let's go to the next one. I want to just say that after many years of work, our lab and several other labs have figured out that Norrie disease, Norrie disease arises from a defect in a protein which sends a signal. So a little bit like VEGF. It's called neurin. That's the name of this protein. It's shown in pink here in this high resolution structure. It binds a specific receptor on the vascular cells, and it instructs them to grow appropriately within the retina. So perhaps no surprise then if this protein shown in pink here, neurin, is defective, which is what is wrong in people with Norrie disease, that there's a problem with vascularization of the retina. We now know that people with the more modest defects, the ones that have just a partial defect, have defects in the receptor or a related, we call it co-receptor molecule. Now, this has led, as shown on the next slide, two biotechnology companies, one called iBio and the other called Surrozen, to engineer molecules that mimic Norrin. Now, these could be used potentially to treat people with vascular developmental defects of the kind that I've shown you, but actually it's being used at this point to treat individuals who have, and I should say in clinical trials, not as an approved drug, in clinical trials, individuals who have excessive leakage of the kind that I showed in the patient with diabetic macular edema, because it turns out the same pathway controls the integrity of the blood vessels and their leakiness or non-leakiness, their tightness to fluid flow. And these molecules will activate the same pathway that the Norrin ligand activates, and it will restore the barrier properties, the tightness, the non-leakiness of the blood vessels. So whether this will generalize in a large patient population, we don't know. It's in early clinical trials. And I'll just say as a disclosure, I'm a scientific advisor to iBio, but it will be known in a couple of years when these large trials read out, and if it succeeds, we hope will be useful to those many patients who are not successfully treated with the anti-VEGF drugs. So I'll stop there, and at the end we'll have some questions. Thank you.
Thank you very much, Dr. Nathans, for that wonderful talk. I'm now pleased to introduce our second speaker, Dr. Jean Bennett. And as a personal anecdote, when I was moving from graduate school back into the clinic and thinking about ophthalmic genetics, this was around the time that the gene therapy clinical trials were starting, and the very first clinical paper I wrote was a review of Dr. Bennett's wonderful work and journey into the gene therapy space. So Dr. Jean Bennett, MD, PhD, received her PhD in zoology and cell and developmental biology from the University of California, Berkeley, and her MD from Harvard Medical School. And she completed fellowships at the University of California, San Francisco, Yale University, and Johns Hopkins University School of Medicine. Dr. Bennett joined the faculty at the University of Pennsylvania School of Medicine in 1992, where she was a professor of ophthalmology and cell and developmental biology until July 2022, when she became the F.M. Kirby Emeritus Professor of Ophthalmology. She continues to co-direct the Center for Advanced Retinal and Ocular Therapeutics, or CARET, at UPenn, while simultaneously further developing gene therapy applications in conjunction with private foundations and biotechnology companies. Dr. Bennett was a scientific lead of a team that translated reversal of blindness in animal models to demonstrate efficacy and safety of gene therapy in children and adults, and her team was the first to enroll pediatric subjects with a non-lethal disease as a gene therapy participants. They completed a re-administration study, and with Spark Therapeutics, of which she's a scientific founder, a registrational gene therapy trial for congenital blindness, the first randomized control multi-center gene therapy phase 3 therapy trial targeting a genetic disease. This work led to the first approved in vivo gene therapy for inherited disease in the US, in Europe, and now in multiple continents. Dr. Bennett established a from bench to bedside program at CARET using her experience in moving successful laboratory findings through rigorous safety and efficacy testing and, if warranted, into clinical trials. And Dr. Bennett continues to develop gene therapies for inherited and acquired retinal diseases and tackling some of the limitations of current gene therapy technologies. She mentors scientists and physicians at all career stages, serves on a number of advisory boards, and is an active member of nonprofit patient-oriented foundations. She's an internationally recognized expert in gene therapy and has received numerous awards. And she is also elected to the American Academy of Arts and Sciences and the Association of American Physicians, the National Academy of Medicine, and the National Academy of Science. And I'd like to introduce Dr. Jean Bennett, and looking forward to your talk.
Robert, thank you so much for that kind introduction. And I'd also like to thank Brian and Diana and all the other supporters of Research to Prevent Blindness. It's an honor to share my thanks and my enthusiasm about what this organization does. It has supported my department as long as I've been there, which is more than 3 decades, and has supported my research and the team that does all the research. And it's more important now than ever to have this kind of support. So shown here is Lancelot, the first dog to benefit from retinal gene therapy when he appeared at Congress for the third time in 2001 to talk about the importance of biomedical research. And this research started in rare disease, a form of Leber's congenital amaurosis, which really gave proof of concept that gene therapy could work in the retina in particular, and has led the way to develop gene therapies for more prevalent diseases, as I'll describe during my talk. So in the next slide, what is gene therapy? It's a technique to harness DNA or RNA to treat or prevent disease. Any product that can be encoded by DNA or RNA is fair game. But for a chronic disease, such as a blindness that starts early in life, you need long-term expression of the gene in the appropriate cells. And you need to know what cells should be treated as well. How do you deliver a gene therapy reagent? Well, I started working on this actually when I was working in a collaboration with Jeremy Nathan's lab and making transgenic mice that shown on the upper left is injecting DNA into the nucleus of newly fertilized egg, where you can then let the egg grow, develop, divide, and that gene can be expressed in numerous organs of the animal. Well, obviously, that's not something we want to do with humans, but we want to deliver the reagent directly to the cells that are in need, in our case today, the retina. And it's impossible to do injections to all those cells. Just think there are 120 million rod photoreceptors in the human retina, 6 million cones, 4 million retinal pigment epithelium cells. How are you going to inject every single one? So one way to do it is to harness viruses to do what they do best, which is to dump their contents, their DNA or RNA, into the cell and deliver the gene that way. And adeno-associated virus, shown in the lower left, is really efficient at delivering genes safely and durably to neuronal cells, such as those that are in the retina. And then if you combine that with the surgical techniques shown in the slide to deliver that adeno-associated virus carrying the new gene into the subretinal space, where it is adjacent to the cells which need the treating, either the retinal pigment epithelial cells underneath or the photoreceptors above, you can get very stable gene transfer. Shown in the lower right is an image of a monkey retina that had been injected 2 years earlier with a adeno-associated virus which carried green fluorescent protein. And you can see those cells glowing green 2 years later with the histologic view just above that. So AAV in the next slide has been used for a number of different diseases, and I'm going to show you more about the retinal pigment epithelium 65 or RP65 disease, of which Lancelot was a beneficiary. That the RP65 encodes an isomeral hydrolase that is present in the retinal pigment epithelium. Without that being there, there can be no vitamin A derivative, the 11 cis that's shown in that upper right, to be supplied to the retina, so the cells can't see. And the children and the dogs with this disease, not only can they not see well, but they also have these abnormal rotatory eye movements known as nystagmus. So in the next slide, one aspect of this disease is there's also slow degeneration. But what you see, we'll start with the slide on the figure on the lower bottom. The uninjected eye is illuminated with light. The pupil does not constrict, whereas the eye on the other side, which had been treated, shows a brisk pupillary light reflex, showing the eye is responding to light. And what is above there is the dog on the left was not treated. He walks around bumping into things, whereas after treatment, he walked around just fine, avoiding objects, even wagging his tail. And shown on the lower right, some of them could read. Not really, but very cute dogs. In the next slide, you can see what happened with the first child in our gene therapy study. He walked into the hospital using his blind cane and holding his mother's hand because he couldn't see. And then a year and a half after one single injection in one eye, he's doing clearly visual things, hammering things. He goes to school, rides his bike to visit his friends, play sports. He was on a little league team, and doing things which he definitely could not do before the gene therapy intervention, and playing board games and living the life of a normal 9-year-old boy. Just to let you know, that was almost 17 years ago. He's all grown up now, has a job, still visual, and enjoying visual activities. In the next slide, one of the challenges we faced in getting approval for this drug through the clinical trials was to select the appropriate outcome measure. In other words, how do you determine whether something is clinically meaningful? And that clinically meaningfulness is one of the metrics of the FDA. Well, at the time when we started, there was only one outcome measure that was used for approval, and that was improving by 3 lines on the eye chart. That was not going to be an appropriate measure for somebody who could not even see an eye chart. So we developed what we call the multi-luminance mobility test, where someone is judged on both the accuracy and speed with which they navigate through a course with arrows, avoiding obstacles that are in their path or to the side of the path. And it's graded on how well they perform at different levels of light that we encounter during a typical day. And in the next slide, you'll see the outcome of these measures. This is at baseline. One subject, representative, starts walking through the course and immediately goes off the course. He's walking very tentatively, kicking the way people who are visually impaired do because they don't want to trip over things in their way. He gets re-guided because he went off course. He's again kicking to feel objects. And if simultaneously you could play the video showing what his performance is like at the year 2 at a much lower light level, you can see a vast difference. Again, at baseline, he's going off course, kicking tentatively. And at year 2, he's walking the course accurately, following the path, going up steps, down steps, avoiding obstacles that are in his way, and finding his way to the end of the path without any problem. And so this was the uniform result in the majority of patients and led to approval of the reagent, which in the next slide you can see is called Veretagine naparvovec Rizzle. They add a 4-syllable consonant to the end of these products, which doesn't spell out anything. This was approved in 2017 in the United States, and then the following year in the European Union, and it's now available nearly worldwide. Surprisingly, the FDA and other regulatory authorities approved it for patients as young as 1 year of age, whereas we had asked approval for 4 years of age, reasoning that it'd be better to allow people to develop normal vision early on and maintain it. So hopefully that is being maintained. And in the next slide, that is one of the questions. So how long does the benefit last? Shown on the lower left is another patient at baseline going through the multi-luminance mobility test, again, tentatively. And then if the test is started at the 9-year point, which you see on the far right, you can see that this person is still navigating accurately and quickly through the course, particularly compared to how they performed at baseline. So 9 years and counting, we are hoping that this benefit lasts for the life of these individuals. We will see as time progresses. So in the next slide, so this has created a path for developing gene therapies for other forms of congenital blindness, other forms of rare blindness. And we got a lot of clues about how to do this from studies in basic biology. Shown on the left is a algae. It's actually called Chlamydomonas, which has flagella. These are cilia which move. And there were numerous studies in this organism which showed, it pinpointed what the molecules were which are important in cilia, and also those that allow transport of proteins up and down those cilia. It's called intraflagellar transport. These have really interesting structures shown in cross-section on this slide. And many of the proteins that are in these structures have been shown to be important for the movement of Chlamydomonas, as well as for cilia, which are found in every single cell in the developing human. And some of these cilia become very sophisticated organelles. Shown in the middle is the modified cilium of a photoreceptor, which has structure very similar to what you see at the base of the Chlamydomonas flagellum and many of the similar proteins. And these proteins allow transport of molecules to the outer segment. Ciliopathies can not only cause blindness, but also severe disease in multiple other organs, such as lung, kidney, etc. So in the next slide, I'd like to share just some results of early phase clinical trials. This one was a gene editing clinical trial that won the top 10 clinical research achievement award recently for its publication of a phase 1 study. This is a study run by Editas and run by Eric Pierce at Mass Eye and Ear. And my colleagues here at University of Pennsylvania participated in treating the 2 children who were enrolled at the end of this study. And what is really interesting to me about this study is that this takes a protein which is called a gene editing protein, a CRISPR-associated protein 9 or Cas9, for which Jennifer Doudna and Charpentier won the Nobel Prize in 2020, and allows editing to occur at discrete places in the genome. In this particular disease, there's a very common mutation which prevents the protein from forming. And what Editas and these colleagues, these investigators did was to use Cas9 to splice out this mutation and allow normal CEP290 protein to be produced and to function, shown as the molecule that was packaged into AAV to allow this to happen. So in the next slide, you can see some of the results. And I'm just showing the results of the 2 children who were enrolled at the center at Children's Hospital of Philadelphia. The untreated eyes on the left and the treated eyes on the right. And in both of these individuals, there were huge gains in light sensitivity. This shows the plot of light sensitivity, and the 2 children are shown above this horizontal line, which shows the area where one would have normal sensitivity. In other words, a huge improvement. And one of them showed extraordinary improvement in visual acuity, improving by 10 lines on the eye chart. And they improved in numerous other measures as well, and some of the adults did as well. So this was particularly promising. In the next slide, importantly, there was no harmful immune response caused by introducing this Cas9 protein, which, remember, is a bacterial protein, which is really quite remarkable and shows also one of the benefits of using the retina as a target for gene therapy. There were some little mounds that were apparent on cross-section using OCT, the same technology that Jeremy Nathan showed you earlier. And these disappeared without any treatment besides some local steroid treatment, leaving no harmful damage. And I'm not going to go over the humoral response data because that was also really benign. In the next slide, there's another disease which is undergoing phase 1 clinical trials, which is also rare, but a different ciliopathy. This one is caused by a gene defect called LCA5, and it's a rare one, but it's one of the most severe early onset retinal degenerations there is. Shown here is a cross-section, a scanning EM of a mouse model of this disease. You can see the outer segments sticking out, looking down at the surface. But in the LCA mouse model of this disease, they don't form. Whatever forms is really misshapen and not functioning at all. In the next slide, you'll be able to see results of the early results of the first 3 adults that were treated in this study. And I'll just show you the first one here. You can see this is pupillometry, and where you see these lines that are green that move to the left, that shows a huge improvement in pupillary light reflex. Here you can see a huge improvement in full field light sensitivities. And remember, that was also an outcome measure for RP65. And this is an outcome which was identified in all of the 3 adult individuals who were enrolled in this program. In the next slide, by microperimetry, there was also improvement shown here. This is the untreated eye. And where it shows a fixation plot down below, and the eye does not fix closely on anything at baseline, but after injection, you can see where the green is localized. There is much better fixation and much more sensitivity. And my pet dog agrees, as you can hear. In the next slide, I'd like to switch to how gene therapy is moving from orphan diseases to more common diseases. And you heard from Dr. Nathans earlier about neovascular diseases and how anti-VEGF therapies have really played a huge role in helping patients see better. However, those interventions require injections into the eye every 6 weeks. And that's not something that everybody wants to do. There are several clinical trials in progress where a gene therapy, the antibody to VEGF is packaged in an AAV and delivered as a one-time treatment by gene therapy. And these trials seem to show good response, good safety. They will read out in another year or 2. So we'll be able to find out more about how well this works. There also.
Dr. Bennett, you're at the 18-minute mark. You should take 2 minutes to wrap up.
Okay,
please.
perfect. Okay, so I'll move to the next slide. And there's also now treatment for end-stage retinal degeneration, which is agnostic as to the exact gene defect which causes the disease. And there's 7 different trials looking at approaches to deliver optogenetic therapy. Shown here are results of one of them, which I won't show because of the time limitations, but they're promising. Time will tell. In the next slide, there are a number of challenges in terms of large genes, which are being approached by different companies and different strategies, being able to splice together messenger RNA, for example, or using 2 different vectors. And if you can move forward with 2 clicks, that would be great. Those are the companies. And AAVs with advanced properties are now being used in retinal gene therapy trials. some of which seem to have better spreading abilities and able to target more cells going from a certain location. One goal that investigators have is to be able to develop gene therapy reagents that can be delivered in the office by intravitreal injection and then home to the cells in need of treatment in the retina. So in the next slide, I'll just move to the end to conclude in the next slide. How to accelerate the breakthroughs? There are a number of ways where I think we can streamline regulatory requirements and natural history studies and develop new approved endpoints, and to develop models which are nonprofit or have open INDs so that for people with really rare diseases can get help that they need quickly. And in the final slide, I just like to express my excitement about the opportunities that there are now for developing treatments using gene-based approaches. These studies in children and rare diseases have led the way and have transformed the health and well-being of those children and adults and provide promise to others. And I'd like to once again thank RPB for leading the way in helping to bring on the new treatments.
All right, thank you both for these really wonderful talks. We have some questions in the chat that we'll get to. And, I just wanted to say that I think this just really nicely highlights that ophthalmology has really led all of the fields in gene therapy, and in large part because of the wonderful basic science that permitted understanding the pathogenesis of these diseases. And so in a lot of ways, ophthalmology is informing all of the medical specialties on how to proceed with gene therapy from a virus and regulatory and everything. So congrats to you both on these really wonderful talks and your wonderful research programs. Okay, so looking into the chat, the first question that we have is there a relationship between genetic eye disease and sibling birth order? And if you need a medical geneticist, I'm happy to chime in too.
Rob, why don't you take that one?
Okay, so for genetic eye disease, genetic diseases are due to changes in the DNA, and sometimes they can be brand new to the person, and sometimes they can be inherited from parents, and those are inherited through the gametes, the sperm or the eggs. And because it is a truly random process, whether you have a new mutation or whether you have a variant that's passed from a parent, it does not depend on sibling birth order. Sometimes when families do find that their first child has a genetic eye disease, that often prompts an important step of seeing a genetic counselor or medical geneticist to really understand what's the risk to future children. But great question. Okay, next question. Speaking of dogs and their eyes, are dogs colorblind?
Maybe I should take that one. Dogs have two kinds of cone photoreceptors. Humans have three. So dogs do have a reduced form of color vision compared to what we humans have, or most of us humans. There are some humans who have only two, and we typically refer to that as, quote, color blind, but really they still have some color sense. They're not living in a black, white, and gray world. But dogs actually are typical of most mammals. Humans are the exception. Dogs, cats, cows, mice, almost all mammals have just two kinds of light receptors for color vision. And we higher primates, gorillas, chimps, humans, are the exception.
Thank you very much. And now a question over to Dr. Bennett. Are there any advances in LCA caused by CRX?
At this point in time, there is just there's laboratory research on CRX. CRX is a hard one to develop a strategy for because it's a developmental gene. It's what's called a homeobox gene, which helps control the differentiation of cells in the retina. so that if one were to develop a treatment, it would have to be fetal, which is not being done yet in humans. It can be done in mice and other animals, but it will take a while to figure that one out.
So much. All right, now a question for Dr. Nathans. Is there a good way of quantifying vascular leakage when imaging on angiography machine versus fluorescence? Or sorry, angiography machine with fluorescence?
I think most ophthalmologists do it sort of semi-quantitatively. It's not rigorously quantitative. I will say that there is an optical coherence tomography, an OCT technology, OCT angiography, which looks at the movement of blood. It does it without a dye. It's completely non-invasive. And that may eventually supplant much of what we're doing with the dye-based methods. And the OCT angiography has the potential to be more quantitative.
Thank you so much. And one more for Dr. Bennett. Do you see the field moving forward addressing IRDs one gene at a time because it's a very slow process? And any genetic treatment available for Fuchs corneal dystrophy?
Yeah, so I'm hoping that the field will start to move much faster. It has been one gene at a time, starting with IRDs, just to de-risk the whole process and to understand what we need to do in terms of dosing and so forth. But I think the field is getting more comfortable with delivering genes to the retina, using AAV in particular. And I think things will start to move faster. I think we still need to de-risk different strategies to make sure they're safe. For example, the gene editing strategy needs to go slowly until we're sure that it works. But yes, I hope it goes faster. Then as far as the Fuchs dystrophy, that disease is a corneal disease, and it is possible to deliver genes through recombinant viruses to the inner aspect of the cornea. However, it's more complicated in terms of getting inflammation and so forth. So stay tuned.
Yeah. And there's one really nice comment here as well as I just want to thank you for the many years of basic science research at LASA Foundation for specialized research in clinical trials. Thank you for all your work. Thank you for that nice comment. And then we'll get back to some of the questions in the chat, but I'm also just curious, Dr. Bennett, Dr. Nathans, do you have any questions for each other now that we're able to have this dialogue today?
I'm happy to take questions from the audience.
Wonderful, great. All right, and so next, could you elaborate on the potential for gene agnostic therapies? And I think that could go to either or both of you.
Gene, you should take that one.
I'm really excited about the potential of gene agnostic therapies. I mean, that would address the question earlier that was raised about, are we going to go one gene at a time? And there are many people who still don't know their gene defect and are waiting to have that identified so that directed treatment can be made. And by the time that is identified, they may not have any cells left to treat. So there are multiple different gene agnostic strategies that are being tested in the clinic now and have shown really great success in the laboratory. So I'm hoping that those will be fruitful.
Rob, I'll just add one thing, which
Sure,
is,
please.
I think a lot of people in the retinal degeneration field think that if we could slow the disease, even if we can't completely prevent it, so something short of an absolute cure, that could be extremely valuable. Many of the disorders are slow degenerations, and we could potentially get years or perhaps even decades of additional vision, and that would be a huge success.
Absolutely agree. And Actually, I do have a question. I think both of your talks struck for me that these diseases also teach us a lot about the underpinning mechanisms of biology, and that biology is also important in other diseases like age-related macular degeneration or even things like cancer. And Dr. Nathans, you had talked about VEGF, which is also used in treatment of new vasculature, both in AMD as well as in cancers. Do you think that your work in the NORI pathway might be extended to age-related diseases or to the general cancer field at some point, or is it already on its way there?
Yeah, Rob, that's a really good point. I think a number of neurologists are looking at this as a potential treatment in the context of blood-brain barrier breakdown. So leakage of the serum contents in the retina, which we see in age-related macular degeneration and diabetic retinopathy, diabetic macular edema, are really not that different from the kind of breakdown that one sees in various neurologic disorders, Alzheimer's, multiple sclerosis, even head trauma. If you clunk your head, there is a local breakdown of the blood-brain barrier. So one question is whether that could be treated by, say, a transient administration of this sort of drug, and during the time of, say, post head trauma, during the time when the leakage is really clinically significant, could we minimize it with a treatment like this? I think eventually that will be tried, and we'll see what happens.
And just to expand on that, I think it might be possible to develop gene therapy in the retina that could then be applied to other organs. So, for example, ciliopathies, CEP290 causes retinal degeneration as well as kidney disease and some lung disease, defects in the brain. And there are other diseases which affect the brain, and I'm sorry, the retina and the cochlea. And so it might be possible to extend treatments to other organs through gene therapy.
You both. And in the last couple minutes, I'll try to get through questions as much as we can. But if not, I'm sorry about that. But this is a question that's directed about CRX, but I think I'll maybe make it more general and just, what do you think about the all the work in stem cell and tissue transplant, and how that relates to gene therapy and things like stage of disease or types of disease?
I can start. I think I think the stem cell field is really interesting. I think it's behind the gene therapy field right now because it's more challenging. It's more challenging to think of how these cells are going to differentiate in the right way, the right manner, and then synapse with the appropriate cells. It's certainly with some cell types, it might be a lot easier to transplant them than the others. But I think it's really exciting, and there's a lot of promise, and there are some clinical trials that are underway or are going to be underway soon. And I'd also like to add that there are prosthetic approaches for treating blindness, which are also interesting.
So our time has
Okay.
come to a close, and we want to be respectful of everyone's time, but I would just want to thank all of you for an excellent session. Dr. Hufnagel, thank you for your able moderation and not only handling questions, but answering some and asking some. Dr. Nathan, thank you for an excellent, very clear overview, and for just a glimpse of your groundbreaking research in Norrie disease. And thank you, Dr. Bennett, for your succinct summary of your pioneering and life-changing work. We're really fortunate to have two such eminent and distinguished scientists as presenters today. And I just want to thank everyone also who participated as an attendee today, and say that there will be a link to this Lunch and Learn that will be made available to everyone who was a participant, either as a speaker or as an audience member. We ask you to really disseminate that to your networks. These things take on a life of their own afterwards. I know we put it on our YouTube channel, and we have many people who download the event after it actually happened. So again, thank you all, and have a good day.